It's my pleasure today to introduce the Grand Round speaker, Dr. Karin Rodland. Dr. Rodland is a researcher in the Integrative Omics at the Pacific Northwest National Laboratory, or PNNL, in Richland, Washington. She received her bachelor's degree from Hood College in Frederick, Maryland, and her PhD from Syracuse University in New York in biology. She then headed to the West Coast, where she's been ever since. First as a visiting assistant professor for about 6 years in Reed College in Portland, Oregon, and then as an assistant, then associate professor at Oregon Health and Science University in Portland. Since 2001, she's been involved in research at the Pacific Northwest National Laboratory in Richland, Washington. Her research focuses on the application of a systems biology approach to interpretation of high-throughput proteomics and genomic data in order to both discover and validate potential biomarkers for cancer, and also to gain mechanistic insight about the dysregulation of normal processes in cancer. She also serves as director of the Precision Medicine Innovative Co-Laboratory, where she coordinates collaborative interactions between Oregon Health and Science University and the PNNL researchers. She works in integrated omics, data science, and multimodal imaging. She's a member of NIH's NCI's CPTAC team, which is the Clinical Proteomic Tumor Analysis Consortium, also the Moonshot and Early Detection Research Network. A little known fact about Karin is that she's a certified licensed pilot. So rather than driving the 4 hours between PNNL in Richland, Washington, and Portland, Oregon, she flies her plane. And I'd like to thank Karin for taking the risk that her flight tonight back to Washington State will depart, and she's not taking her own plane. It's Delta. Our lecture today is entitled Proteogenomics and Pathways, Functional Insights from Integrated Omics. So Karin, thanks for coming.
And I have flown over Minneapolis many times on my way to Oshkosh, Wisconsin, which is the AACR of little airplanes. Just so that you know that. Well, thank you for having me here. This is a picture of lovely PNNL on the Columbia River in central Washington. It is not in Seattle. And it's summer, so it's green. And in the rest of the year, it's the desert. So let's see. Why proteogenomics? So a lot of research has been done on the genomics of cancer. The Cancer Genome Atlas has done whole genome sequencing and RNA-seq and has come up with some very good insights as to what is driving various cancers. But we all know that cancer and cell function is really executed at the protein level. And so what we wanted to do is determine whether adding proteomics to genomic analysis could help you prioritize all the genomic changes that you see. And what we have here is our first task is ovarian cancer. And I want to point out that ovarian cancer is really a disease of disorganized chromosomes. There is the P53 mutation that you see in all serous high-grade ovarian cancer, and then after that, there's no one driving mutation. Everything else is at about 14%. So you can't just say this is a RAS-driven tumor or this is an EGFR-driven tumor. You really have to look at a variety of pathways. So the first effort we published in Cell 2 years ago, and it's an integrated proteogenomic characterization of human high-grade serous ovarian cancer. What we actually did here was we got the same OCT blocks that TCGA had used. And so we had all the genomic and RNA-seq and methylation information, and we did proteomics and phosphoproteomics on those blocks. So my next slide is why ovarian cancer. I probably don't have to tell you folks, I'm sitting in a oncology department, but ovarian cancer is really a disease of chromosomal instability. How do you pick out of 113 focal copy number aberrations the one that's really driving the disease? This is not BCR-ABL. This is not TMPRSS2-ERG. There's too many of them to figure out what's going on. The other reason why focusing on ovarian cancer is it's the poster child for biomarkers. It always has been the poster child for biomarkers because 5-year survival is 90% or better for stage 1-2, and 17 to 40% for stage 3-4, but only 20% of women present in stage 1-2 when they are diagnosed with ovarian cancer. Now, it could be that these are completely different diseases, that high-grade serous ovarian cancer is a disease of the fallopian tube that has migrated to the peritoneal cavity, and that stage 1-2 is really a disease of the ovarian surface epithelium. But we won't know that until we do the genomic and the proteomic and the phosphoproteomic analyses. So those are the questions that are framing this study. So the experiment was proteogenomics. CPTAC, as just Amy said, stands for the Clinical Proteomic Tumor Analysis Consortium, and it's nationwide. There's a center at the Broad, there's a center at Johns Hopkins, there's a center at Nashville, which is now kind of moved to Baylor. There's Pacific Northwest National Lab, and then there are some ancillary sites. And there's a combination of proteome characterization centers that are mass spec facilities that really specialize in doing the in-depth, high-throughput analyses, and proteogenomic data analysis centers, NYU, Vanderbilt, Baylor, Michigan, Washington University, St. Louis, and they have contributed to the genomic analyses. So for this first study that I'm going to talk about, Johns Hopkins and PNNL both opted to do ovarian cancer out of the short list of tumors that we could choose to work from. And the target was to do 100 tumors at each site. The actual end number turns out to be less because of the quality of the specimens. You sometimes can't do what you want to do. Because of my interest in ovarian cancer and knowing how bad the outcome, the prognosis is in this disease, I deliberately picked the extremes of survival in the TCGA data set. So I picked women who died in fewer than 2 years, and I picked women who were still alive or alive more than 10 years. So we did a short surviving, long surviving comparison. Because we did not have appropriate normals to do the comparison, TCGA used PBMC DNA for the germline DNA. They didn't need to have normal ovarian. And that leads to the whole question of what's the right control for ovarian cancer anyhow. Hopkins was interested in BRCA-ness, and they wanted to find a surrogate biomarker for BRCA-ness, for the inability to repair DNA damage. So they chose BRCA1-2 mutants and then homologous repair deficient competent, so HRD minus. Then it's a double negative, but homologous repair competent cell lines. So we had 84 tumor samples, 32 overlapped with Hopkins. So my personal emphasis is on information flow. This is the central dogma of molecular biology that I taught to my students at Reed College years and years and years ago. And we all know it's more complicated now. We know that the flow of information from DNA to RNA to protein is modified by non-coding RNAs, proteolytic processing and degradation, methylation and epigenetics. So now the question is, what should we measure? What should we really measure if we want to know what's going on? So what we did measure in our ovarian cancer study was proteomics and phosphoproteomics. This is a graph of each of our 82 samples and what kinds of data we had on it. So we had copy number variants on every sample. We had DNA methylation on almost all samples. We had RNA-seq on half of the samples. We had transcriptional analysis by microRNA analysis on most of the samples. We had RPPA, which is a form of proteomics. It's an antibody-based form of proteomics. It stands for reverse phase protein arrays. And it's based on about 200 well-characterized antibodies, of which about 30 to 40 are for phosphoproteins. Now, one of the things you'll see is how much richer our data set was as we go along. But we had those data for a comparator, and then we had microRNA data. We also had the clinical metadata, the initial diagnosis, the follow-up diagnosis, and the overall survival. So that was the input to the study. We wanted to integrate the genome and the proteome. That's the whole objective of this study. So we started with copy number variations, mRNA, protein, phosphopeptides. we did correlation analysis. So there are two basic ways of integrating these types of data. One is based on known relationships in the curated literature. Many people do that. They go to KEGG, they go to genome ontologies, and they look for known relationships. We were doing an unbiased de novo approach where we looked for statistical correlations between changes in copy number, mRNA abundance, and protein abundance, and phosphopeptide abundance. And theoretically, that way you can come up with some novel observations. And we then did a lot of gene set enrichment analysis on those things that were correlated, because we believe in rolling it up to pathways. So if you focus on function and correlation, you actually simplify the complexity of the data that you're looking at. They call, the informatics people call it reducing the dimensionality of the data. Instead of looking at everything as a point, you want to look at the patterns. And so this is a diagram of the CNA protein correlations. And so for any locus on a chromosome, we looked at the copy number variants on the chromosome and the abundance of the proteins that were encoded at that locus. And so this nice red diagonal is effects in cis. If we had an amplification of a locus, we saw more protein. If we had a deletion of a locus, we saw less protein. And you can see that in ovarian cancer, you have very strong cis effects. Not all cancers had this strong a cis effect. If you take a look at the colorectal cancer diagram out of Vanderbilt, same analysis, they had much less of a cis effect. So copy number variants, by and large, get translated into protein in ovarian cancer. But these vertical lines are a locus on a chromosome where the abundance of proteins at other loci on other chromosomes was significantly altered in trans. And we thought that this would be very interesting, looking at a higher level of regulation. So there were hotspots of trans regulation. And when we took and did the gene set analysis enrichment on the proteins in a vertical band and translated it here to the hotspots, we found that if you rolled it up to functional levels, we had an upregulation of pathways associated with proliferation, motility invasion, and immune regulation. These are 3 of the 10 hallmarks of cancer. And so what we're trying to do here is ask ourselves, for ovarian cancer, what are the most important hallmarks that are driving the disease? Because they will tell you something about potential therapies to develop for treating the disease. So proliferation came up quite strong, as well as motility and immune regulation. We found that the functional analysis was more predictive, more informative than a simple molecular cataloging. So if you took the RNA data and you did a heat map, which everybody in TCGA did all over the place and loved doing it, you could come up with 4 different RNA subtypes, and we came up with 5 different proteomic subtypes. We found a new subtype that we called stromal that was very enriched for complement cascade, angiogenesis factors. It probably reflected highly vascularized tumors. And you can see that the transcriptome-driven subtypes, there was no difference in survival. Perhaps there's a little bit worse survival in these highly vascularized tumors. But we took we took this data on the proteins affected in trans, and we did an analysis based on the survival, our short and long survivors, and we came up with a multivariate signature at the protein level for short versus long survival. So here's our long survivors, here's our short survivors, and very high p-value. This is the 95% confidence interval around the curve. So this looks promising. What we are currently in the process of doing is a second set of prospectively collected ovarian cancer samples, an independent cohort. This includes women from Poland and from Thailand, as well as North American continent. And we don't have the outcomes data really to validate this, but we are looking at the women who died within the first year of entry into the study, and we're finding that the short survival signature was increased in these women compared to the women who are still alive after a year, and we found that the normal samples have an even higher rating of the long survival signature. So it's looking promising. Now, people often ask about mRNA-protein correlation. Why bother to measure protein? This is the standard Bible with the transcriptomic people, and I hope I'm not stepping on anybody's toes here. But, you know, if you can measure the RNA, why measure the protein? We got that old dogma. Well, for most of the mRNA and protein, the correlation is pretty good. But notice we actually have a tail of negatively correlated mRNAs and proteins. And so, and we saw this exact pattern in every tumor type we looked at. We saw it in colorectal, we saw it in breast, we're seeing it in endometrial, we're seeing it in every cancer type that we're looking at. And if you take a look at the functional groups, if you do a waterfall plot of the most highly correlated mRNA and proteins and the most least well correlated mRNA and proteins, you find that they actually separate out by functional groups. And in a not surprising way, things like ribosome function, OXY4, mRNA splicing, complement coagulation, there's not a really good correlation between the mRNA and the protein abundance. But if you look at what we used to call immediate early responses, things that are upregulated quickly in response to perturbation, we find a good correlation between mRNA and protein for interferon responsive genes, nucleotide metabolism, amino acid metabolism. And again, this functional correlation, this grouping by functions, was consistent across colorectal cancer and breast cancer. So we all know that there is post-transcriptional regulation of RNA translation and protein stability. It is driven by the function of those genes. So that led us to our driving hypothesis, and our driving hypothesis is that if we studied things at the pathway level, it would be more informative than if we studied it at the single gene or transcript or protein level. And so what we want to do is integrate the multiple data types, compare the data sources at a pathway level, and get a better understanding of the mechanisms contributing to survival. Now, this is perfect. Bear with me, this is my cute little story about why we care about pathways. And it couldn't be a better day for this. So this is the route map for United Airlines. This is the genome of United Airlines. This is everywhere you can go from point A to point B on a United Airlines jet, right? This is the potential. This is a clip from FlightAware. And FlightAware is a flight tracking app, many of you probably know. And this is the actual phosphoproteome of United on a normal day. This is where airplanes are going. They're going back and forth to Chicago. There's some around Denver. There's some down here around Houston. There's a lot of activity going to New York. So this is the phosphoproteome in health. This is the normal United Airlines. And so then here's a day like today. This is Winter Storm Jonas when I put this together. And, you know, we have a little bit of a disruption here. Here's the phosphoproteome in disease. Notice that it's not just that nobody is going to New York anymore because there's snow sitting over it. Notice that nobody's going through Chicago anymore. Everybody's going through Denver. And this is my point, is that when you perturb a system, you don't just perturb one point, you perturb the whole system. We have to look at the whole thing. So. And of course, I have to get back home, and I'm looking at the phosphoproteome of disease, so it's going to be interesting. So we are very much focused on phosphoproteomics. I'm a signal transducer from way back. It's, you know, information flows by the transverse phosphate from protein to protein to protein. So the first thing we found is that if we just took our short versus long survivors and did a plot of the extent of phosphorylation, that all of the short survivors were just at an increase in phosphorylation in general compared to the long survivors. And then if we mapped it by pathways, we could see here long survivals are in blue, short survivals are in yellow. Short surviving people are enriched for all these pathways with phosphorylation: PDGFR-beta, VEGF, cytokine CXCR4, CDC42, RAC, estrogen receptor and isophospholipid generating pathways. So we get a very strong distinction between short and long survivors at the phosphopeptide level. This was using 4 different types of data and ranking the most highly differentially abundant pathways between short survivors and long survivors based on phosphorylation. In purple, global proteomics, that's the abundance of the protein regardless of modification. mRNA and copy number variants didn't drive anything here. And you can see that we got much more information out of phosphorylation than out of the global proteome, and only 2 of our pathways really popped up at the mRNA level, if you just looked at them individually. And so, oh, I did this backwards. This is the RPPA data on protein abundance and protein phosphorylation for the same patients. And we could see STAT and MEC and BRAF and SARC. But the problem with RPPA data is you can only see the proteins that they put on the chip. So if you look at the global phosphoproteomic data and the global phosphopeptide data, this is the picture that you get instead. And we can really see the PGFR beta pathway completely upregulated. I know this is a complex slide, but every protein that we could see is in a purple box. If the mRNA was unchanged, the arrows go this way. The mRNA was increased, the arrows go this way. If the total protein abundance was unchanged, the arrows go this way. If the total protein abundance was decreased, down arrows. mRNA was decreased, down arrows. And then the little P's and black circles are the phosphorylation levels. And again, red arrows are increased, green arrows are decreased. You can see that almost all the information content in this pathway comes from the phosphorylation events and the increase in the phosphorylation. And one of the things that I need to point out, you can see that we looked at protein abundance and at phosphorylation abundance. Phosphorylation was normalized to protein abundance. So if we say that phosphorylation was increased, it wasn't just because the number of STAT5A molecules was increased, but the proportionate phosphorylation of STAT5A was also increased. And so that's another thing that you can do with phosphoproteomics that's much more difficult to do with Western blots. There's no direct comparison between this. So one of our thoughts for clinical relevance is, you folks all know that bevacizumab has been tried in ovarian cancer in unselected patients and showed no advantage in survival. What if we took the CML pathway and we only gave bevacizumab to the women who had upregulation of the PGFR beta pathway? Would we see an increase in the survival of those women who actually had the upregulated pathway that was being targeted by the drug? So that's precision medicine, precision oncology. That's where we're going with all of this. That is the clinical application that we're driving towards. So, and here's an example using links data of how one would go about doing this. So these are breast cancer data where we have links data on breast cancer epithelial cells, memory epithelial cells, and how they responded to different inhibitors. And then we have individual patients and the response of a patient's cells ex vivo to each one of these inhibitors. So if you take a look at this particular patient here, you can see that a deep blue means responsive and a light blue means resistant. So this woman was highly responsive to these 5 agents, and we can now go in and prescribe for this woman specifically. And that's obviously the whole concept behind precision oncology. So the inhibitors that we saw, JAK, GSK3, DOT1 ligand, gamma secretase, and Notch. So those were the types of things that you would come up and give a try with this woman in that kind of trial. So that takes us to the second project that we're working on now, the precision medicine application. And so this is the standard version of precision medicine. You take actionable mutations, maybe you do some in vitro phenotyping, you throw it all into a black box analytical engine, and you come up with a treatment recommendation. So the cells were responsive to these 2 drugs, resistant to these 2 drugs, so we're going to give the woman pertuzumab. That's how it works right now. So there's some limitations, though, to precision oncology, the way it's being practiced like that. First of all, you have to have an actionable mutation. FDA has now, woken up and seen the light, and drugs are now approved on the basis of the mutation they target rather than the tumor type of the clinical trial. So you can use trametinib against anyone with a BRAF mutation. So, and here are some of the actionable mutations that we have drugs for: BRAF, NRAS, ALK, ABL. BRCA1/2 mutations, or neoantigens for immunotherapy, which is still kind of a actionable mutation. The initial response rate is high. It's very encouraging, but we're still seeing resistance develop. We're still seeing very high levels of resistance developing, whether it's a targeted therapy or whether it's just plain old cytotoxic therapy. So how can we solve the resistance problem? And then if you have multiple mutations, which therapy are you going to pick? That's definitely a problem. So the folks that I am collaborating with at Oregon Health Sciences are doing what they call smart treatments. Everybody's got their name for it. It's the same kind of thing. This is serial measurements of molecular and architectural response to therapy. So they're looking at mutations, transcriptome subtypes. They're doing 43-color immunohistochemistry. So yeah. They bleach and then they do, and then they bleach and then they do it again. I saw you shaking your head. So they can do about 6 or 10 at a time. And they're doing immunophenotyping and pathway activities by mRNA and functional screens in vitro and PDXs in mice. Well, what we want to do is add proteomics and phosphoproteomics to this mix so that you're actually getting a direct readout of the pathway information rather than transcriptional signatures. And so, to me, the advantages of proteogenomics are they're truly personalized. You're not just dependent on driver mutations. You're looking at the way that mutation is being manifested in that particular patient. It adds a direct measurement of pathway activity, and it provides very deep coverage, more than 43 colors, more than 200 RPPA antibodies. And so it's more unbiased. And it provides a lot of information on post-transcription regulation. It's going to push our basic science understanding of the disease as well. So back to CPTAC, the new part is the translational, does it matter? Translational research centers, and they have a preclinical arm and a clinical arm. We had to pair up with an NCI-funded clinical study and get actual patient specimens and start to tackle the problem of drug resistance with actual patient specimens. So the preclinical arm was to establish a signature of drug response in preclinical models and then validate the signatures in retrospectively collected patient samples. Then the clinical arm was to tie yourself to an ongoing clinical trial and test how good your predictions based on proteomics and phosphoproteomics were compared to the other arms of the clinical trial. So we paired up with Brian Drucker at OHSU for acute myeloid leukemia. Brian and I had adjacent labs back when I was back at OHSU, and I watched the whole imatinib Gleevec story evolve. So this is a great partnership for us and has just worked out really well. So we're trying, what they are doing with AML is they're doing ex vivo IC50 assays to predict the small molecule inhibitor for the AML patients because there's a large number of mutations in AML. We're taking the same baseline data from those patients and seeing if we can predict the response in the ex vivo assay. And then we are also able to validate the signature in the primary patient samples. And we also want to look at the dynamic response of the patient samples to drug treatment. So it's not just the baseline, but how do they actually respond. And when they have cryopreserved viables, we can actually do a real cell biology experiment and treat the cells and look at the proteome and the phosphoproteome. So those are our objectives. So our initial proof of principle study, can you even see differences? These are 3 of the 5 cell lines that the FDA requires that they use in their ex vivo assay. So in order to get FDA approval in the clinical trial, they have 5 cell lines. They have to use any 3 of these 5 because they know how they're going to respond to specific drugs. So K562, which is actually BCR-ABL, THP1, which is a RAS mutation and responds well to trametinib, and MOM14, which is a FLT3 ITD mutation and responds well to sorafenib. And this is the proteomics, and this is the phosphoproteomics. And you can see it, they broke out. You can't mistake one of those cell lines for the other. They have very different signatures. K562 is very highly upregulated in proliferation-associated genes and phosphopeptides, which would go well with the activated ABL oncogene. Both THP1 and MOM1 are more highly regulated in differentiation-associated, immune function-associated proteins. There's this odd upregulation of metallothionein-related metallothionein proteins in THP1. Don't ask me why that's going on, but a fair amount of overlap. So we thought that this was encouraging. We also did something that's called kinase substrate enhancement analysis. So we look at the phosphorylation of known substrates of a particular kinase, and we back infer the kinase activity from changes in the phosphorylation of known substrates. That's KSEA. And we looked at ABL1 substrate phosphorylation in these cell lines, and wow, look at the difference in K562 ABL substrates compared to MOM14 and THP1. So it's validating this approach for inferring the enzyme, the kinase that's driving the phosphorylation changes. We saw similar changes in CDK1, CDK2, and JNK. So this is a very promising technique that we're doing. Go ahead. So then we really were interested in doing the cell biology experiment with patient samples. We started with the cell line samples, and of course we need to know what's the best time point to pick. And the shorter the time point for our clinical application, the better, because what we would love to do, okay, so I believe that someday there is going to be a CLIA-approved mass spec sitting in a little room off your OR. It can happen. And but I'll settle for a nanostring assay or whatever after we determine what's important. But you're going to be able to take a sample out of the patient, treat it with the drugs, hopefully for 30 minutes, through it through the mass spec, and the next day you're going to get an answer back and you're going to know what drug to put the person on. That's my dream, we can all dream. So we had these 4 cell lines, K562 with BCR-ABL, treated with dasatinib, MOM14 with the FLT3 mutation, treated with sorafenib, HL60, which like THP1s are mutant RAS, and we treated them with trametinib, and CMKs, which are JAK3 mutated, and we treated them with ruxolitinib. And I now can pronounce those, which I couldn't before. And here's what we found, some very interesting observations. First of all, we saw definite changes at half an hour. These are all compared to the baseline. So we see responses as early as half an hour, and basically at 3 hours, the response is pretty much similar to what we saw at half an hour. So if you see it at half an hour, you'll probably see it at 30 minutes. And then we get a return to baseline at 16 hours, or return towards baseline. The temporal pattern changes from protein to protein. This is protein abundance, and this is phosphoproteomic abundance. ABL is a really beautiful one, and we don't need to work with that. But here's FLT3 that we're going to end up focusing on, and you get some really nice changes at half an hour. So we're hoping that the half an hour time point will really work here. So, and we're busy using machine learning to generate a signature out of these data. We need to run more patient samples because you have to have large N in order to get there. But so we now went on to look at actual patients. And these are patients in the Beat AML 1.0 trial, which is It's not an NCI-sponsored clinical trial. It's sponsored by the Leukemia and Lymphoma Society. We kind of had a fight to get NCI to accept it. The condition was that all the data had to be publicly available, and they are. Actually, the results of Beat AML 1.0 were just published in Nature a month ago, and so all of the data is publicly available. So here are 16 patients that we pulled out, and the mutational status for each one of them. You can see that FLT3-ITD is the most common mutation, but you can see again that there's a lot of different mutations in AML that are known driving oncogenic mutations. We had drug response data for every one of the patients, so we could compare our predictions from protein and phosphoprotein with the measured drug response data. And this is the result of our first experiment. So this is baseline. This is not treated. This is proteomics and phosphoproteomics. These are the 16 patients that we had enough yield to do phosphoproteomics. Bad news about phosphoproteomics is the way you do the analysis, we have to have about 800 micrograms of peptide to start out with because phosphorylation is such a rare event compared to total protein. We're working on solving that problem. And so here's the mutation for each patient and whether they were sensitive to a drug or resistant. And the interesting thing here is that the patients do not cluster by mutation, but they do cluster by, in general, whether they were sensitive or not. So it looks like there's a common way in which to be sensitive to small molecule inhibitors, and the genes that are upregulated in this quadrant are tyrosine kinases and receptors associated with immune function, plasma membrane receptors associated with TLR receptors and things of that nature. So it's almost like if you're a well-behaved, proliferation-primed tumor, you'll respond to the small molecule inhibitors. If you're less well-behaved, you'll be resistant. We're really working on analyzing these at present. So we do have a couple of NCI-approved clinical trials. And in this case, we're looking at, we're doing the inhibitor screen on either newly diagnosed AML or relapsed refractory, picking a treatment based on the IC50 assay, and we're going to do baseline proteomics on all these patients. If they do respond in vitro, then they get that inhibitor, and if they get a response, then they're fine, they stay on that. If they have progressive disease on day 28, they go back to get a second-line therapy. We get post-treatment proteomics from the patients. So, and we're not going to have a lot of samples, so this is driving our technology development in terms of decreasing the sample size that we need to do this. But this is the clinical trial. And the only difference between this arm and that arm is one's newly diagnosed, and so the patients are also on conventional chemotherapy, 7+3, and then these are relapsed refractory, so they're just getting the small molecule inhibitor. That's the difference between those two arms. So, okay. Some of the preliminary work that we've done on patient samples that supports the idea that we'll be able to do this is pacritinib as a test case. So pacritinib is an inhibitor. It inhibits JAK family kinases, KIT, CSF, and that makes it an interesting drug to try in the context of AML. It's got a high degree of specificity for IRAK1 as a kinase, and it also has specificity for JAK2, FLT3, and CSF1R. And what we did in this case was we did deep phosphoproteomics on, 8 patients that we knew whether the patient was sensitive or resistant to pacritinib based on the ex vivo assay. And we did the phosphoproteomics, and you can see, we have a really nice clustering of sensitive patients and resistant patients, and you can kind of see a change in intensity of phosphorylation of these genes, RPS6 kinase, ADRBK1, CDK7, in the most resistant patients. We also did an analysis for common phosphorylation motifs in the resistant patients. when we saw these increases in phosphorylation, or these increases in phosphorylation, was there a common motif that was being targeted? So we found an ERK1, ERK2 motif, and we found a GSK3 motif. So again, these might be ways to feed this information back to the guys who are developing the inhibitors so they can develop more specific inhibitors. That's the type of thing that we're hoping to do. And so P38 MAP kinase substrates were very highly correlated with sensitivity to pacritinib, and this is the type of thing that we're going to be doing going forward, looking for specific inhibitors and their targets. One real problem is, what do you do when you have multiple mutations? How do you pick a drug? So this is, individual mutations and the other co-occurring mutations at the same time. So you see most patients have more than one mutation. The traditional way to do it is empirically. That's what they're doing in the ex vivo assay. They're doing combination of drugs and looking to see whether it's additive or non-additive when you combine more than one drug. And then if it's more than additive, a CR less than one indicates that the combination is more effective than either single agent. What we want to do is actually, so what you end up as a result of that is this kind of really complicated matrix and very difficult to understand. We really want to understand the crosstalk between pathways so that we can intelligently decide how to do a combination therapy. So we're doing it based on pathway analysis. We have a program called Causal Path that will take a look at the protein, phosphoprotein data and map it back to known curated pathways. So right now we're applying that to a clinical trial of drug combinations. Ruxolitinib and venetoclax are being used in combination on some of the AML patients. It's part of the Beat AML 2.0, and it's been approved. And so we're looking at patients who respond and patients who don't respond. And that work is in progress right now. So it's actually in the mass spec. Can't report on it yet. So the take-home message from proteogenomics is really that we can, I didn't show you those data, but in our first analysis of the TCGA papers, we can see at the protein level splice variants and fusion proteins and mutant peptides that were predicted from the genome data. So we search our proteome data against the individual's own genome, and we can see some of those events. They're difficult to detect, and we don't know whether that means that there's a bias against translating them or whether it just means that they're difficult to detect. And that's one of the weaknesses of mass spec. The trans effect of copy number variants on protein expression was something that we did not expect. We did not expect it to be as strong as it was. And it looks like another way, another level at which selection is operating on clones, so that you're selecting for proliferation, you're selecting for immune regulation. There are many, many ways of doing that, of increasing the proliferative rate of a cell. Evolution doesn't care which mechanism your clone is using. It's selecting for clones that are going to proliferate more rapidly. And then if you combine proteomic, transcriptomic, and genome data, you can really identify the subnetworks. You can identify what is driving a particular patient's cancer, and then you can use that information to recommend therapies in addition to the simple process of a mutant RAS or a wild-type RAS. And so we focus on pathways. And that is the end of the content of this. I want to thank all the people who've worked with me on this project. Proteogenomics is really a team science. We have quite a repertoire of talented individuals at PNNL. Paul Pichowski and Tao Liu are my analytical chemists who run the mass spec and come up with clever ways to decrease sample requirements, increase sensitivity, all those things. Jason McDermott and Vlad Petyk and Sam Payne are my bioinformatics guys who interpret and analyze the data for me. Dick Smith is the Thomas Edison of mass spectrometry. He just keeps inventing new mass spectrometers and new front ends to increase their sensitivity, and I give him something to aim for rather than just inventing something. So it's a great relationship. And then the OHSU team, Brian Drucker, Jeff Tyner is an associate professor who's highlighted the ex vivo assays. Uma and Mark are the clinicians. Anna Priya is working with Jeff on the signal translation side. And then Christina and Samantha and Chris just do everything. So thank you very much for your attention, and I'd love to take questions.
Do you have any questions for Dr. Rodland?
Yes, next slide is
I just want to go back to the work on ovarian cancer, and I understand that this was, as you mentioned, all tumor types, and I don't think, you may have mentioned something about stage, but I didn't hear that.
I'm sorry. And that's my bad, because it's in my head. These were only high-grade serous ovarian cancers.
Thank you.
Stage 3 and 4.
Thank you.
That's one of the reasons why doing the heat map clustering was an exercise in futility, because we've already down selected. We don't have mucinous, we don't have clear cell, we don't have endometrioid. So I think that these are far more homogeneous at the mRNA level than, say, the PAM50 panel for breast cancers, where you have the luminal and the basal. And I had to fight with the transcriptome people on that the whole time. But yes, it's only high-grade serous.
Makes sense. Thank you.
You're welcome.
I have a
Yes.
somewhat related question to that too. For those tumor samples that you were taking, what was the anatomic sites? Were they omentum metastases, peritoneal metastases, were they actually from either the fimbriae or the ovarian primary? Because even within TCGA, there was the identification of different transcription patterns, subtyping, if you took a patient's matched primary versus...
They were all, we selected them to be all omental. Yes.
So, couple of questions. First, did you find congruence between RPPL data and your data? And second, have you done the quality control where you take a sample here and you take a sample 5 centimeters away, and you get the exact same results? Have you done spiking? Basically, I'm coming from the single cell approach here, wondering how much, if you have 5% infiltration of immune cells, does it throw off your proteomic?
Okay. Yes, we get concordance between RPPA and our phosphoproteomics and proteomic measurements. We see there are times when we see different phosphorylation sites than the ones that they think the antibody is directed against with the RPPA. So if you look at where we assign the phosphorylation, it may not be the conventional one, and we don't know whether that's real or whatnot. But at the protein level and was this protein phosphorylated, we see excellent 86-88% concordance between our results and our PPA. We do spike in experiments. If we have a signature, we validate the signature with targeted proteomics, which is based on stable isotope labeled synthetic peptides for what it is that we want to see. And so for the papers, the key associations, we do a targeted proteomics analysis to make sure that we've not misidentified the protein. As far as your infiltration of immune cells and whatnot question, that's an excellent question. And I try to make it very clear to NCI program and to people. When we do these bulk tumor analyses, we are analyzing the tumor as a tissue. We are not actually analyzing the epithelial component that has become malignant and comparing it to the normal epithelium. That's not what we're doing. We're not doing laser capture. We're looking at doing that going forward, but these experiments with TCGA, they gave them to us as cryopulverized, macro-dissected blocks. So when I say there's a 5th subtype that's a stromal subtype that has a lot of blood proteins in it, I tell you, I think what I'm looking at is a highly vascularized tumor or a highly vascularized region of the tumor. 5 centimeters away, it's going to be completely different. In this next round of analysis, we're doing cores. So we're doing 1.8 millimeter cores through the tumor block, and we're doing cores that we think are epithelial enriched and cores that we think are stromal enriched, and we're doing multiple cores of the same type. And epithelial enriched cores do cluster together compared to stromal enriched cores, but it's not 100%. It's 60%, it's 50% correlation. Plus, when you're doing a core through the block, it's not a layer cake, it's a layer cake that's done. And my son's a geologist, so he knows exactly what I'm talking about, you know. And so we think we're getting epithelial all the way through, but we're doing multiple slices for H&E, and we're not. It's really tough. Yes, Scott, and then you.
I was fascinated by your talk. Thank you so much. And you talked about having mass specs outside of every operating room. And I was trying to visualize how this is going to apply to community hospitals where most ovarian cancers are taken care of. And I actually have a related second question. Could you talk a little bit more about the informatics support that you need to get the proteogenomics data that you have?
So the informatics support is crucial. That's the differentiator between success and failure in these. I'm lucky to have a strong team at PNNL because we've been doing these kind of proteomic studies for 15 years and developed. And then we have access to very strong people who are genomics informaticists at Wash University, at NYU, who are now adding proteomics to what they're doing. In terms of the clinical translation, I know there's a lot of steps, and in actuality, what's probably going to happen is that our informaticists and our mass spec people are going to identify the signature, and there will be some simplified assay that can be run by a med tech type person in a community hospital. And it could be antibody based, but I could also see a very simplified mass spec setup with the synthetic peptide spiked into it, and you just, you had a cocktail. You mix this with that, and you stick it through the mass spec, and this setting and that setting, and it comes back out. But yeah, that's going to be what it's going to be. That's an engineering problem. I was married to an engineer for 33 years. They can fix things. I'm very confident that engineers can fix things. Scott, you had a question.
Yeah, so it was really interesting that you saw that global shift towards increased phosphorylation in short versus long-term survivors. So what's driving that? You saw some pathways that were associated with that shift, but it's unclear whether those are the cause or the consequence of that. So what's flipping that switch towards increased phosphorylation globally?
I don't know what the prime driver is. Whatever it is, let's say that a large percentage of phosphorylation-associated pathways are proliferation-associated, DNA replication-associated. So whatever is the fundamental process, and it might be P53, that's pushing proliferation in these cells, it's upregulating all these pathways in parallel rather than just one. So we're seeing it across the board.
So you think it's like a DNA level event that's then translating?
Yeah, and these cells are proliferating like crazy, so every single pathway that's associated with proliferation and DNA replication and transcriptional activation and protein quality checking, they're all upregulated, they're all cranked up, the accelerator is pushed forward, so everybody's going 90 miles an hour instead of 50.
So and then just by extension as well, has anybody looked to see whether there's a global shift in lipid phosphorylation or sugar phosphorylation as well?
So we're just now adding metabolomics to these studies. We do have metabolomic capabilities at PNNL as well. We're just now adding those. In colon cancer, the Baylor group has got a real nice story about metabolism-associated proteins being upregulated in the microsatellite stable subtype of colon cancer. So the microsatellite unstable, that's driven by the instability, but in the microsatellite stable, either the metabolism is a driver or the metabolism is a consequence. But at the pyruvate lactate ratio, which doesn't surprise anybody. Yes.
I love your discussion of dreams and we all need they're also nightmares too. Do you, this would be way down the road, do you see a point in time where insurance companies would demand something like this in order to pay for the use of certain very high expensive drugs?
Yeah, I can see the insurance companies wanting to have this kind of basic science backup for the drug. And then if it's a drug that's only going to be selectively successful, I could see that they want the companion diagnostic. And again, the FDA is going this way. Almost all of the new small molecules are coming up with companion diagnostics. What you want is not to have one companion diagnostic for one drug and have to run 50 assays. What you might want is one global assay that you can look at it and say,
That's it. Or ones.
yes.
So it's similar question, general question about what's the concept. I really like the idea of precision medicine in general, but before that we were all very interested in evidence-based medicine. And it's important to appreciate that the two are almost mutually.
If you this is actually a pet peeve of mine. So if by evidence-based medicine you mean that the largest number of people in the population will respond to this treatment without any knowledge about their molecular defects, then they're incompatible. But if by evidence-based medicine you mean, is there a rational fact-based explanation for the response or not response, they're completely compatible.
Oh, I agree with that. But get big enough group of patients.
That is a problem.
With a certain way. And
Yes,
slice it so
that
thin
is
everybody
the
can-
huge problem.
How are you ever going to get
Yeah,
the
no, I totally agree with that. So if you're insisting on population-based measurements, and every precision medicine patient is an N of 1 or an N of 5, you've got to change the standards of evidence, which means you have to change the statistics. So in order to get this adopted, we're going to have fundamental changes in applied math and statistics so that you can have confidence in your low end results.
Companies FDA.
Okay, well, thank you very much.
You're welcome. Sorry for the burst of energy there, but
John's gonna get his thing on here.
I'll just pull it out because it's not really going to hurt it, even though it tells me it's going to hurt it.